Okay, good morning, everyone. So today we have a collection of presentations regarding metals as they're related to certain diseases. And so today we're going to have the first presentation done by Alondra and Saomi. So I want either one of you to go ahead and start projecting your presentations.
Yes. Can you see?
I can see it.
Okay. Actually, hold on 1 minute. My headphones are not connected.
Not a problem. Sami, are you presenting?
I can see it.
Yeah, I can see it. I was just wondering if you were actually talking because I couldn't hear anything.
Okay, well, I'm gonna start.
Yeah, go ahead.
Good morning. What?
Go ahead. Yeah, you're good.
Okay. Good morning. Today we're going to have 3 seminars. The first one is going to be about the levels of metals and metalloids that affect the severity of disease, COVID-19 being the case of study. The second one is going to focus on the levels of metals and proteins as indicators of Parkinson's disease. And the last one is going to be about examining metal regulation in brain-related pathologies such as tauopathies. Next slide, please.
Is there a delay here?
I think Xiaomi is having.
Okay, you guys, I can go ahead and project for you.
Yeah, that would be great. Thank you.
Okay, let me go ahead and do that, okay?
I'm sorry about that. I was having.
Don't worry about it. That's the whole reason why I wanted the presentation. Thank you. Go ahead.
Okay, so my name is Alondra Velez, and together with Xiaomi Torres, we're going to present the scientific article named Associations of Essential and Toxic Metals and Metalloids in Whole Blood with both Disease Severity and Mortality in Patients with COVID-19. So this study was done at Wuhan, China, and 306 COVID-19 patients participated. They were hospitalized from February 10 to March 15, 2020. Okay, so let's start by talking about COVID-19. First of all, coronaviruses are a large family of viruses that can infect both animals and humans. They are named for their crown-like spikes in their surface, and as you can see in the picture in the right, COVID-19 is an infection disease that's caused by a newly discovered coronavirus called respiratory syndrome coronavirus 2, or SARS-CoV-2. It was first identified in 2019, and the most common symptoms are cough, fever, and tiredness. However, you can experience shortness of breath, body aches, sore throat, loss of taste or smell, diarrhea, headache, fatigue, nausea, vomiting, and congestion. Also, there can be patients who are asymptomatic. So the global burden of infection of COVID-19 is really high. These are the disease statistics. As you can see, in Puerto Rico, there are total cases of 140,000 with 2,112 deaths. The big impact that this disease has had on us can be observed when you see the worldwide statistics. There has been a total cases of approximately 127 million with 2.78 million deaths all around the world. Because of this, the need for identifying the factors to reduce the risk of infection is a medical priority right now. So the nutrition's role. It is well established that nutrition plays a very important role in immune function. The abnormalities of micronutrients could be associated with increased infection risk of COVID-19. Because of this, some studies have been done, and a wealth of clinical data has shown that vitamins and trace elements play important and complementary roles in supporting the immune system. Some of these trace elements are metals and metalloids that will be studied in this article. Next slide, please. So, as I was saying, some of these trace elements that are essential are metalloids and metals. First of all, metalloids are elements whose properties show intermediate properties between the metals and solid non-metals. These, together with some metals, are key part of our nutrition. For example, iron, zinc, magnesium, selenium, and copper are essential micronutrients that have shown important immune properties that include the regulation of inflammation, T-cell proliferation and function, oxidative burst and self-protection, immune cell proliferation and movement, antimicrobial activity, cell-mediated immunity, and antibody production and function. Also, iron specifically takes a big part in the production of hemoglobin and myoglobin. So the author's main objective was to analyze essential minerals and heavy metals in whole blood of COVID-19 patients and to investigate their association with the disease's severity and outcome. As I said before, 306 COVID-19 positive and hospitalized patients participated in this study. First of all, the patients were classified between non-severe and severe group. The non-severe group included mild and moderate cases that presented symptoms such as fever and respiratory symptoms with or without radiological findings of pneumonia. The severe group included severe and critical cases with symptoms such as respiratory distress and failure requiring mechanical ventilation. All of their outcomes and specific characteristics are summarized in Table 1. It is important to know that all of the patients they came from the same social background. However, they were divided by age and sex. As you can see, from the total of 306 patients, there were 148 patients who were male and 158 female. Another key thing that was evaluated was the outcome of these patients. As you can see from the non-severe group, the non-severe group was of a total of 202 patients, and all of them recovered. However, when you look at the severe group, from a total of 104 patients, only 89 recovered and 15 patients deceased. Together with other serious medical conditions, as you can see in the next slide, this slide is la continuación from the table. And as you can see, together with serious medical conditions, other blood routine indicator tests were made. As you can see below, albumin, pre-albumin, blood cell counts, and hemoglobin tests were done. This is because previous studies have shown that they are associated with inflammatory processes that have to do with COVID-19 evolution. So in order to carry out this study, 2 milliliters of blood were collected in vials for trace elements. The samples were analyzed by quadruple inductively coupled mass spectrometer, and the preparation of the calibration standards was done. Once they had the calibration standards, they diluted 200 microliters of blood samples and quantified the essential minerals by ICP-MS with a quality assurance.
All right, so I'll be discussing the results of this study. Starting off with the levels of whole blood metals and metalloids in COVID-19 patients. These results were obtained from inductively coupled plasma mass spectrometry, as Alondra just mentioned. For each element, the reference range, medium for all patients, medium for non-severe patients, and medium for severe patients are presented in the table. The first 9 elements, from copper to cadmium, only exhibited concentrations above the limit of quantification. The limit of quantification being the lowest concentration of an analyte that can be quantitatively detected. The bottom 3 elements exhibited concentrations below the limit of quantification. Only 1.62% of the measured chromium had concentrations that were lower than the limit of quantification. The same is true for 24.18% of the measured mercury and 54.9% of thallium. Most, if not all, of those concentrations below the limit of quantification were due to the measurements of patients with the non-severe cases of COVID-19. We can also see that the median of chromium for all patients does surpass the established reference range. Copper, calcium, and chromium exhibited higher values for patients with severe COVID-19. The rest of the elements, excluding cadmium and mercury, exhibited lower values. And if we move on to the next slide, we can see the levels of whole blood metals and metalloids in recovered and diseased patients with severe COVID-19. The diseased patients exhibited higher concentrations of cadmium and chromium and lower concentrations of arsenic compared to the recovered patients with severe cases of COVID-19. Here we can also see that the interquartile age range, for the diseased patients has higher numbers, meaning more older people did not recover. And more than half of the diseased patients were male. In the next slide, we can see the longitudinal dynamic profiles of the 12 metals and metalloids in whole blood. Patients with severe cases of COVID-19, which are shown in red, had lower levels of magnesium, manganese, iron, zinc, arsenic, thallium, and lead compared to the patients with non-severe cases, which are shown in blue. They also had higher levels of calcium, chromium, and copper compared to the non-severe COVID-19 patients consistently throughout the weeks. In the next slide, we can see the correlation matrix among the whole blood elements. The numbers above are Spearman's correlation coefficients, and the numbers below are p-values. The color blue represents a negative association, and the color orange represents a positive association. We can see that the majority of the correlations between the elements were positive, since the majority of the squares are orange. The darker the color, the stronger the negative or positive correlation. For example, iron and zinc, which you can see is the darkest orange square, have the highest positive correlation out of all of the other elements. And another example is that iron and calcium have the strongest negative association. In the next slide, thank you. In the next slide, we can see the correlation matrix between the whole blood elements and laboratory parameters. The results show that iron and hemoglobin have the strongest positive association. That means that as hemoglobin increases or decreases, so does iron, and vice versa. And this is to be expected, right? Calcium and hemoglobin exhibit the strongest negative correlation. Therefore, if hemoglobin increases, calcium decreases, and the opposite is also true. And we can also see that the majority of the correlations here are weak and therefore have weak linear relationships. And these are all of the squares in white, which is the majority of them. In the next slide. All right, so in this slide, we can see the risk factors for severe COVID-19 illness that were established by the researchers. Age, sex, and whole blood iron concentrations are all independent factors that can predict the severity of COVID-19 in patients. People over the range, or over the age, of 65 are 4.19 times more likely to experience a severe case of COVID-19. Men are 1.97 times more likely than women to experience a severe case of COVID-19, and people with less than 376.67 milligrams per liter of whole blood iron are more likely to experience a severe case of COVID-19. In the next slide, thank you. In the next slide, we can see the risk factors for fatal outcome in COVID-19 patients. The fatal outcome of a COVID-19 patient can be predicted by independent factors such as whole blood chromium concentrations, whole blood cadmium concentrations, and the comorbidity of cardiovascular diseases. Patients with 1.32 or more micrograms of chromium per liter of blood are 9.595 times more likely to die of COVID-19. Patients with 0.68 or more micrograms of cadmium per liter of blood are 12.734 times more likely to die due to COVID-19. And patients that suffer from cardiovascular diseases are 6.91 times more likely to not recover from COVID-19. And the next slide is the conclusion. And basically, this study successfully analyzed the whole blood levels of metals and metalloids in a cohort of 306 patients with COVID-19. Blood samples of patients with severe and non-severe cases of COVID-19 underwent inductively coupled plasma mass spectrometry. The results demonstrated that age, sex, and levels of iron in the blood independently affect the severity of the disease, and that levels of chromium, cadmium, and existing cardiovascular diseases affect the fatal outcome of patients with COVID-19.
So continuing with the conclusion, this is the first study focusing on the association of whole blood metals and metalloids with the severity and outcome of COVID-19. Also, it was concluded that nutrition may play an important and safe role in helping mitigate patient morbidity and mortality. It is important to note that several aspects of the COVID-19 mimic metabolic event, such as the drop of T cells. Because of this, constant monitoring of essential metals, such as magnesium, could be a possible strategy to influence disease contraction and progression.
Those are our references. And we want to thank you all for your time and your attention, and any questions are welcomed.
All right, thank you, Zomi and Alondra. All right, so let's open up questions, brief questions to the class. So anyone has a question, please get on your microphone or post it into the chat. And even if we don't respond to it at this moment, we will do so by the end of the class time. All right, so I'm going to start while your class, oh, actually, we do have one. Gemily, chromium and cadmium were higher in the deceased group compared to the recovered group, while arsenic was lower. Why do you think this happens? Great question. Why do you guys think?
All right, so chromium plays a role in immunostimulatory and immunosuppressive processes, and these processes affect T and B lymphocytes, I believe, and also the production of cytokines. And it just so happens that patients that suffer from severe cases of COVID-19 also experience cytokine dysfunctions and lymphocyte abnormalities. Now, this information could possibly serve as maybe a possible explanation for the high levels of chromium in these patients. Now, the higher levels of cadmium and lower levels of arsenic may be due to abnormalities in the vascular system, and this happens with a lot of the elements that are covered in the study. In addition to that, specifically cadmium, I believe, exhibits cumulative toxicity in some organs, and I think it has also been found that cadmium has a negative effect on the respiratory system, especially disrupting the function of the lungs. And yeah, I hope that answers your question.
Thank you. So based on your assessment of this paper, do you guys have any suggestions as to potential nutritional suggestions that could be made in light of the data that has been collected, especially some of the possible better correlations for survival?
Can you repeat the question? Sorry.
All right, so basically, from your own assessment of the paper, do you have any suggestions that can be made that's on the level of nutrition, as far as based on those metals that seem to have a favorable factor for survival or for the lesser severity of the disease?
Yeah, I think voy a contestar en español. Aumentando la cantidad de específicamente manganeso, hierro y zinc en la nutrición de las personas, yo pienso que eso sería bien beneficioso, porque se pudo demostrar que quizás está relacionado a la incidencia de la enfermedad y la mortalidad. Así que quizás sería un factor que se puede explorar más, hacer más estudios para entonces mejorar eso.
All right, thank you. Okay, so any additional questions, please post them in the chat room. While you guys do post them in the chat room, so I do want people to participate in that way, we're going to move on to our next presentation. So thank you again, you guys, for that presentation. All right, so we're going to move on to Kiara and Gladimaris. If you guys can go ahead and start sharing your presentation.
Yeah, I need you to stop presenting, Professor, so that I can start
I'll do
presenting.
that. Okay. Sometimes my screen gets a little bit,
No worries.
there we go.
Okay, can you see my presentation?
Yes, I do.
Perfect. Well, my Kara is gonna start presenting first.
Okay, well, good morning, everyone. Today, my partner Gladimaris and I, Kara Gonzalez, will be presenting the article Interdependence of Metals and its Binding Proteins in the Parkinson's Disease for Diagnosis. We're going to start with a little introduction about what is Parkinson's disease. Well, Parkinson's disease is a common neurodegenerative disorder that affects over 10 million people. It is characterized by the loss of dopaminergic neurons at the substantia nigra region of the brain. Its symptoms, such as tremors, rigidity, and bradykinesia, may be present. And the main focus of the study is the diagnosis, and the diagnostic for Parkinson's disease is based only on medical history and radiological imaging. This is why they decided to do the study, because there is no molecular markers that are available to detect Parkinson's disease. However, it is known that there is a genetic association and that there are also risk factors such as aging, genetic mutation, and environmental exposures of metals and pesticides, but the mechanism of those metals and pesticides exposures are very much unknown. And this study presents what is the emergent metabolomic approach, which Gladimares will briefly explain.
Well, metallomics is a large-scale analysis of metals in cells, biofluids, or tissues. It is used as a high throughput the techniques such as atomic absorption spectroscopy, inductively coupled plasma mass spectrometry, and inductively coupled optical emission spectrometry. It is used for detecting and estimating the metal concentrations in biological systems, particularly metallomics analysis of cerebrospinal fluid, serum, urine, and saliva has proven useful to identify metal biomarkers for several diseases, which is what they want to study. Additionally, metallomics provide a functional insight on metal interacting molecules such as DNA, RNA, protein, and metabolites. These are the methods that we're going to start
in
discussing.
this article, yes, well, in these articles, they have a variety of methods. These are the very first methods that they used. They started with data collection and protein interaction, later principal component analysis based on network properties. They later continued with clustering, text mining, and meta-analysis, and again, a text mining. I will explain what they were able to find through these methods. So they started with data collection and protein interactions, and pretty much what they did was they collected data from various biological databases, and through this they were able to identify 41 metals that bounded to about 5,000 proteins. They decided to exclude membrane proteins because they're on bounding of metals at their functional active sites. They later proceed to group them the metals, the proteins, I mean, based on the bonded metal that they have. And this way they concluded with 24 metal binding protein groups. And these groups were later subjected to protein-protein interactions to construct metal-protein networks. And to be specific, they focused on 24 metal-protein networks. In the next slide, we can see the metal-protein network of copper. And we can see that the red dots, they indicate the copper binding proteins. So those are the proteins associated with copper. And in total, they have about 4,000 interacting proteins. The purple in this case indicates the interacting proteins. And this is the example of the copper metal protein network, so that it's easier to visualize what exactly is the metal protein network. After this, they decided to determine the biological significance of the 24 metal protein networks. And to do this, they chose 24 at random, 24 molecular pathway networks that were generated and subjected to topological analysis. They selected 7 important topological parameters that were determined for the 72 networks, including the 24 that they had previously decided on, then the 24 random and the 24 molecular pathway networks, and that's the total of the 72 networks, which describe the network properties. In the next slide, we can see a representation of this analysis, which is a principal component analysis plot, and it's based on the topological properties. Here we can see the red dots, and those red dots represent the 24 metal protein networks. The green dots represent the molecular pathway network, and the blue dots represent the randomized network. We can see that the green dots and the red dots are actually grouped together, and this is how they were able to conclude that they did have a biological function, because the randomized ones just didn't even coincide with the molecular pathway networks. After this, they checked the grouping of the 24 metal protein networks with the 24 molecular pathway networks, and as we saw, they were able to group them, and that way they constructed the metal protein networks that are biologically meaningful. And with this, they created the protein hubs, and the protein hubs pretty much represent the core functional components of the metal protein networks. They were 24 overexpressing hubs in total, but they chose 18 of those because they had an association with Parkinson's disease, and they were able to determine that by finding a minimum of 1 protein that was related to Parkinson's disease in each of those hubs. And the next part of the method, Gladys Maris will explain it.
On this part, we're going to be talking about other methods like meta-analysis, classification algorithm to classify metal proteins, network hubs, sample collection and processing procedure, analysis of hub-associated metals in serum and cerebrospinal fluid, gene expressions, analysis of cluster protein antioxidants, and other markers. Meta-analysis is to determine the regulation of the selected hub that Kara mentioned in Parkinson's disease. The expression of proteins encoding genes in each hub was subjected to a meta-analysis, for which the microarray gene expression datasets were extracted from NCBI geodataset and expression omnibus GEO. 18 hubs from the Parkinson's disease in brain and blood were retrieved from the NCBI GEO datasets to determine their regulations. Here we have the brain gene expression that were retrieved from the datasets, and here we have the blood gene expressions that were retrieved also. And the collected microarray datasets were subjected into an integrative meta-analysis of gene expressions, also called as INMEX, and the analysis was carried out by the following procedure from one of the researchers. The gene intensity was log transformed, and Lima proceeded to determine the significant gene's p-value. Datasets between the control group and the Parkinson's disease ones. For the collected 14 brain data sets, the meta-analysis was conducted to determine the significant genes in Parkinson's disease and Parkinson's disease with p-values. Similarly, the significant Parkinson's disease gene of blood were identified from the merged 4 datasets. The EBI Array Express datasets included a normal control group and a Parkinson's disease group for the participants in brain and blood tissues. The meta-analysis of 302 proteins encoding genes of 18 hubs were analyzed using integrative meta-analysis of gene expression, considering the primary goal to identify differentially expressed genes between the control versus the Parkinson's disease in brain and blood. Of the 302 genes, as I mentioned before, 75 were significantly expressed, differentially expressed in 39 over and 36 underexpressed in the Parkinson's disease brain, which attributed to 16 metalloprotein network. Similarly, in the blood, 5 genes were upregulated and 86 were downregulated. That attributed to 17 metalloprotein network hubs. The genes of these hubs were mapped with their appropriate gene expression data of the brain and the blood and subjected to classification algorithm. The classification algorithm based on metalloprotein network classification was performed using Weka software. 34 distinct classification algorithms were used to classify each hub into the Parkinson's disease from the normal, based on the merged expression data of the meta-analysis. The protein of each hub was considered as a feature with its corresponding gene expression value as input for classification algorithm. It was evaluated based on 10 times 10-fold cross-validation technique. The classification ability of predicting normal or Parkinson's by each algorithm for brain and blood was evaluated by accessing the output based on sensitivity, specificity, accuracies, and Matthews correlation coefficient. Based on the performance of the top 5 hubs, showing better accuracy between 34 algorithms were selected, and their proteins were subjected to a key EGG pathway enrichment analysis. For example, in brain, iron showed better performance with accuracy ranged between 50 to 91% in all the 34 algorithms, where cesium hub showed least accuracy between 44 and 56%. For validation, the hub-related metal was analyzed using atomic absorption spectroscopy in serum of control in the control group and in the Parkinson's disease. Simultaneously, the protein in each hub was functionally prioritized using a top gene technique. As I mentioned before, of analyzed hubs, aluminum, calcium, copper, and iron, and magnesium, were overrepresented in most of the algorithms commonly noticed in both brain, blood, contributing to a better classification of Parkinson's disease from the normal control group. On a further pathway enrichment analysis of 132 proteins of these 5 hubs that I mentioned before showed the association with wide spectrum of molecular pathway related to Parkinson's disease. Particularly the calcium, copper, iron, and magnesium were commonly regulated 29 molecular pathways. These pathways, they attributed to environmental information processing, immune, neural, and endocrine aging signal transduction mechanism. They prioritized the analysis of protein in each metal hop, suggesting these proteins mentioned here. And they're functionally important in the 5 metal I mentioned before. There were aluminum, calcium, copper, and iron, and magnesium hops.
So what they did after that is that they focused on the metal concentration and gene expression of these 5 hops that Gladimatis mentioned in specific. They decided to measure the metal concentrations in blood serum and in cerebrospinal fluid using atomic absorption spectroscopy. And in this table, we can see a significant increase in the serum of aluminum, copper, and magnesium, and also a decrease in copper and iron. On the other side, when we talk about cerebrospinal fluid, we saw a decrease in aluminum, copper, and iron, and an increase in calcium and magnesium. And that's the difference that was observed between the Parkinson's disease and the control group. After this, they decided to do a quantitative real-time gene expression analysis of the hop proteins encoding genes in the peripheral blood mononuclear cell from control versus Parkinson's disease. And with this technique, they were able to see an unregulation on certain genes. To be specific, they mentioned 7 of them, and most of them showed a significant upregulation in Parkinson's disease, but the only one that showed a downregulated in Parkinson's disease compared to the control group was the SOD1 gene, which is a gene that provides instruction for making enzymes that play a role in how molecules bind to copper and zinc and break down toxins.
For the discussion on copper hop, B2M, and MAP1S were differentially regulated in both the brain and blood tissue, confirming its impairment in Parkinson's disease. B2M is a member of major histocompatibility complex 1, also known as MHC1, was widely expressed in various regions of the human brain. Also, it was established that MHC1 is a neuron plays a vital role in synaptic input, synaptic plasticity, and regulates axonal regeneration. MH1 is involved in cerebellum motor learning and cortical synapses functions. Altered MHC1 expression related to neuroinflammatory mechanisms that implicated the immune-mediated neurodegenerative process in disease, included Parkinson's disease. The B2M, which is related, is part of the MHC1, has a strong association with complex neurodegenerative disease such as Alzheimer's and Parkinson's disease. Protein of copper hop showed a significant involvement in tight junctions, amoebiasis, and MHC, signaling pathways. The tight junctions between the cells contribute fundamental structures of blood-brain barrier, also known as BBB. BBB play a crucial role in maintaining a stable environment for the central nervous system. Altered tight junctions and BBB disruption has been reported in Parkinson's disease pathogenesis. The researchers explain that they used the copper because it was the one who expressed it more, but with other metals that I mentioned before, it also had similar reactions. These metals, the 5 metals I mentioned before, act as a cofactor for several proteins involved in various pathological processes in Parkinson's disease. Of the analyzed hops, the protein of each metals were involved in immunological, neuroendocrinal, and aging process that linked with the Parkinson's disease pathogenesis. as in concussion. Pathway analysis paves a way to understand the role of 5 significant metal hubs in Parkinson's disease. Results were noticed in the study confirming the importance of these metals in serum and the cerebrospinal fluid of Parkinson's disease pathology. Metal interdependency between the cerebrospinal fluid and serum in Parkinson's disease suggested the impairment of metal transport in BBB and BCB.
And before we finish our presentation, we wanted to say that this article helps to understand the role of metals and its binding proteins in Parkinson's disease, and analyzing the levels of serum aluminum, calcium, copper, and magnesium, along with the gene expression, can be useful in detecting Parkinson's disease, which was the main focus of this article. And before we end, we wanted to mention that this was actually the first study reporting the metalloprotein networks to describe the influence of metal binding proteins in Parkinson's disease. And since they were the first study to do this, it's actually it shows promise and as a possible way to diagnose Parkinson's disease. And with this, we conclude our presentation. So thank you for your time, and we are open to questions.
All right, thank you so much to the both of you. So I'm going to take a brief question. So if anyone has a question from the class, please go ahead. Go ahead, Adriana.
Voy a decirlo en español porque se me hace más fácil.
No, go ahead.
No sé si tenga mucho que ver con el artículo, pero pregunto si puede ser posible que, por ejemplo, ustedes mencionaron que la dopamina, pues, tiene que ver con el Parkinson's, el depletion de dopamine en ese circuito del cerebro, que si tal vez los metales pueden interact con la dopamine también y enhance el efecto de que no llegue el dopamine, por lo tanto tal vez promover Parkinson's. No sé si me estoy desviando del artículo, pero no sé si sepan algo de eso.
Pues, te voy a ser bien sincera, Ariana, no había pensado en eso, y como esto uno de los primeros, o es el primero, mejor dicho, el primer estudio que hacen sobre eso, no sabría contestarte la pregunta sobre si podría afectar o no. No sé si Kiara tiene algo que aportar sobre eso.
Lo único que puedo decir es que es un poco complicado porque algo que ellos siempre mencionan en el artículo es que como hay tanto pathways que pueden influir sobre el Parkinson's disease, que en realidad no hay una causa que se conozca que causa y afecta a Parkinson's disease, es difícil decir si afectan o no afectan y cómo lo hacen. Porque son muchas proteínas que están involucradas en cada hub de los metales.
Incluso ellos mencionan en el artículo que al ser ellos los primeros, ¿verdad? También tienen una desventaja en su artículo, es que toda esta investigación es basada solamente en una población del sur de la India y que ellos esperan en investigaciones más adelante usar otras poblaciones del mundo para tener quizás una mayor variación de mappings de diferentes proteínas con estos metales. So, apenas está empezando, pero en verdad es lo único que se ha conseguido hasta ahora, ya que nadie nunca ha podido detectar la enfermedad del Parkinson.
Okay, thank you guys. Adriana, that's an interesting question. So dopamine is technically a catechol, so it does have very, strong metal binding capabilities. So that's an interesting possibility of metal interaction with that particular molecule, maybe ablating some of its activity. So, essentially lowering it, even if its concentration may be still there, but that's something worthwhile looking into, because there are examples of metals interacting with certain substrates of enzymes, and therefore, because of that interaction, serving as inhibitors. So I think that's something that's really interesting to explore. My question for you guys is, why aluminum of the set of hubs, aluminum being something that's not known to be an essential metal. So why that particular metal?
Well, what I read from the
Well-
well, Kara, do you want to answer?
Either or. Go ahead. You both can give a shot.
was-
Go ahead.
Okay, I'll go after Kara.
Okay, well, what I actually wanted to say is that aluminum, even though it's not an essential metal, in order to choose the specific 5 metals, they looked at previously reported data. And since aluminum was a metal which was linked previously to Parkinson's disease, that's the main reason why they also included it, even though it's non-essential.
Yeah, she practically said what I was going to say. So yeah, it's because of the database they used before. And los resultados que obtuvieron era de los más que estaba linkeado con el Parkinson disease, a pesar de todos los otros metales que ellos ya habían estudiado, escogieron los más que se asociaban con la enfermedad.
Yeah, I would say that's the right answer to go for, because there could be or could not be urban legend of aluminum and toxicity and associated with these neurological diseases. So it's a safe bet to go for something like that to explore if there actually is any correlation. So yeah, that seems reasonable to me. All right, guys, thank you very much.
I
So-
see that Gemily wrote a question. If you
Yeah.
want, we can answer at the end.
Yeah, we'll save some time later on. All right, so we'll move on to the next presentation. So Javier, can you go ahead and start projecting?
Professor, can you present the
Okay, sure, I can do that.
the PowerPoint?
Uh,
I send it to you.
yeah, I have it.
Okay.
Let me just find it quickly. All right, well, good morning, everyone, or good afternoon at this point. So I'm actually helping Javier to present this particular article, not the specific article. I'm actually going to give the context. So I'm going to go back to the slide when he is going to present. So just to give you guys a bit of a context, I like to relate things that we've talked about previously. So you have all seen this slide before, or at least I hope you have, if you were paying attention. We talked about Alzheimer's disease, which is technically a type of tauopathy. And we look specifically at what is part of the manifestation of this disease, which is that we see certain proteins undergoing a major change. And so one of those proteins is the APP protein, which is the amyloid precursor protein, which at the membrane undergoes a proteolytic event, which leads to the formation of the A beta peptides, which we discussed previously. Those can go on to then form the amyloid plaques that we've discussed. And then there's also the intracellular protein, the tau proteins. Here's just a representation of them. And so those proteins can become hyperphosphorylated, leading to the formation or the change in conformation and aggregation, resulting in what are known as the neurofibrillary tangles. So you get a very similar manifestation of these two proteins as a consequence of this particular disease. And we talked about a number of hypotheses regarding the potential onset and propagation of Alzheimer's disease, where we focus on the amyloid peptides undergoing these transformations in structure that leads to these really high order formations, these fibrils. And we discuss how metals, particularly iron, iron 2, iron 3, copper 1, copper 2, zinc 2, how those particular metal ions can play a role in the manifestation of these higher order fibril structures, as they can facilitate coordination to the peptides and therefore link one set of structures to another, giving further aggregation. But in addition to that, there is a component of the metals, the redox active metals, the iron and the copper, enabling the formation of reactive oxygen species and even reactive nitrogen species that can induce an oxidative stress, and that oxidative stress can result in the oxidation of biomolecules, and really important intracellular biomolecules that can ultimately lead to neuronal death. But something that seems to be correlated or can attribute to the the manifestation of the phosphorylated tau converting into those neurofibrillary tangles is perhaps the some of the results of the amyloid fibrils. As we talked about previously, the release of some of the reactive oxygen species can potentially reach the cellular environment, and that can contribute to the formation, or at least the further propagation of these neurofibrillary tangles. But something that we didn't really talk about before, but is actually, adds to the complexity of this disease, but really a better understanding of what's going on in the brain, is the iron regulation that occurs between APP protein and tau. So there does appear to be an interaction within the cellular environment, within the neuronal cellular environment, that facilitates a translocation of the APP protein to the membrane, and then that translocation allows for the APP protein to interact with ferroportin. And as we know, ferroportin is a protein that is responsible for the efflux of iron in its reduced form, iron 2. And so that is actually a significant process because that contributes to the maintenance of homeostasis of iron within the cell environment. So obviously, the cell is a simplification. So it's been proposed that soluble tau allows for a lower neuronal iron levels by enabling the translocation of APP to the neuronal membrane. And so without soluble tau, neurons accumulate toxic levels of this metal. Studies have shown with tau knockout mice that they can develop a Parkinsonism, so building off from our previous presentation, with an impaired cognition phenotype that can be ameliorated via iron chelation. So interestingly, one of the components of Alzheimer's disease and also related pathologies is a loss of iron homeostasis by tau. And so here I just give you this perspective of, we already have talked about APP undergoing the proteolytic event that leads to the A beta peptides, the amyloid plaque. Well, there appears to be a correlation between this important kinase, and you guys probably remember this from our discussions of lithium, glycogen synthase kinase 3 beta, which we talked about its capability of contributing to phosphorylation. So there is some speculation that this particular kinase can contribute to the phosphorylation of tau. So here's just the cartoon representation of that. So that modification of that protein can potentially, as we know, lead to the neurofibrillary tangles, but very importantly, it can affect the interaction between tau and the APP. And so what is the net effect of that? Well, the net effect of that is an increased amount of the labile iron in the cellular environment. An excessive amount of iron can lead to oxidative stress. It can lead to oxidation of important proteins, DNA, and ultimately to neuronal death. So it seemed logical, and actually a number of you have asked me this. I remember Adriana and also Christopher have asked about this possibility of, well, what about using lithium for exploring its therapeutic value, because we have talked about lithium serving as an inhibitor of sorts of GSK3 beta, and so maybe it could play a function here. And so you guys are smart, and so people have in fact done this type of analysis. There have been low dosage lithium clinical trials for use in AD treatment, but unfortunately, at least in those trials, the results were disappointing because there was a lack of efficacy. But people are still interested in exploring lithium and the possibility that it can potentially serve a function, at least for the treatment of AD. And so further studies have been done. An in vivo study was done using MRI on humans given lithium, and it resulted in iron level increases in the substantia nigra region of the brain. Lithium treatment of BL6 C57J, or black mice as they're typically known, at doses inducing blood levels below the human therapeutic range. Remember, there's a very limited window for lithium, so they were aiming for low levels. Well, actually, that amount that was treated was enough to result in cognitive impairment and brain atrophy. It's believed that lithium treatment in mice may result in neuronal apoptosis by triggering this particular pathway, the calcineurin nuclear factor of activated T cells, fast ligand signaling pathway. So if you trigger that apoptosis, it can lead to neuronal death, and it can lead to this, then the physiological effects that were observed in those particular mice. All right, so this leads to Javier's paper. Okay, Javier, so you can go ahead and start presenting.
Okay, so as the professor said, a loss of homeostasis in the essential metals, including iron, copper, and zinc, is believed to be correlated with the pathogenesis of tauopathies. Iron, in particular, may play a direct role in the hyperphosphorylation of the tau protein and its aggregation in neurofibrillary tangles. So the aim of this study, I forgot to say the name of the paper. The paper is titled Characterizing the Spatial and Temporal Brain Metal Profile in a Mouse Model of Tauopathy. So the aim of this study is they characterize the spatial temporal brain metallomic, as the title says, profile in a mouse model of tauopathy. The mouse model is RTG tau 4510. And the other aim is to provide insight into the potential interaction between the tau protein pathology, so the neurofibrillary triangles, and iron, and other metals as zinc and copper, but mainly iron. So the methodology, they were using as I said, the RTG 4510 mouse model, which overexpresses human tau bearing the P301L mutation. That is a mutation that in humans causes tauopathy. And they review the spatial and temporal distribution of iron at 4, 7, and 12 months of age. So between that times. This model recapitulates several behavioral and pathological phenotypes that are evident in human tauopathies, such as age-dependent cognitive decline. That's why they used 4, 7, and 12 months to see the cognitive decline, neural loss, hippocampal atrophy, and neurofibrillary tangle formation. So they use two techniques. The first, they use laser ablation inductive coupled plasma mass spectrometry. That is an analytical technique that enables high sensitive elemental analysis on solid samples to characterize the metal in the brain. This was used to investigate the spatial distribution of iron in the brain. And the other technique they used was the size exclusion chromatography mass spectrometry, the second one there. That's another analytical technique that this one enables size differentiation to obtain a better analysis. And this one was used to measure iron bound to other metalloproteins, such as, in the case of iron, ferritin. That is a protein that stores iron. So they use about 5 to 7 quantity of mice, the type of mice I told before, and these mice were euthanized at 4, 7, and 12 months of age. Then they, from the left hemisphere of the cerebellum, the cerebellum, the hippocampus, and cortex were rapidly dissected, and each sample, along with the remaining tissue from the left hemisphere, were stored at 80 degrees until analysis. The right hemisphere, the other, was drop fixed in 4% paraformaldehyde overnight and subsequently cryopreserved in 30% sucrose in PBS. And then the statistical analysis was carried using a type of software named GraphPad Prism 8. In this, can you go back a little bit? Okay, in this picture, we can see the region of interest. This is one hemisphere of the brain of the mice. And in this, this is all the entire hemisphere. In red, you can see the hippocampus, and in purple, you can see the cortex. So, in the results, they found for iron, the spatial temporal distribution of iron accumulates with age in both RTG 4510 and wild type mice. In the entire hemisphere, there was a 52 increase in brain iron levels from 4 to 12 months. In wild type, there was this was in wild type and a 151% increase in RTG 4510 mice. As you can see, there's a lot of difference between the wild type, that was the control group, and the RTG 4510 mice. In the hippocampus, the wild type had a 63% increase in iron, and in the RTG 4510 mice, a 135% increase. In the cortex, there was a 57% increase in wild type and 178% in RTG 4510. In the right, you can see in the image in the A, you can see that the blue parts are low quantities of iron, and if you go to green, then to yellow, to finally to red, that's high quantities of iron. And you can see the differences between wild type and the TG mice. So this is the regional analysis, and it reveals that tau accumulates primarily within the hippocampus and cortical region. So in the hippocampus and the cortex, it accumulates more, and this reflects a similar pathology profile to Alzheimer's disease. So they also not only examine iron, but they also examine iron bound to metalloproteins. So using the size exclusion chromatography mass spectrometry, they revealed 3 peaks associated with iron protein complexes. The first peak, as seen here, is a ferritin complex with iron. As you can see, that's the part that has the most in the brain. The peak 2 is believed to be associated with cytochrome C bounded with iron, and the third peak, they need to do more analysis but it's believed to be other metalloproteins bounded with iron. Here you can see a 56% increase in ferritin bounded to iron from 4 to 12 months in RTG 4510 mice, while the wild type mice was a 24% increase that was not significant. They also examined the ferritin quantities in the brain. The ferritin protein increased in RTG 4510, but in wild type did not increase significantly. The results were 146% increase in ferritin in the hippocampus, 46% increase of ferritin in the cortex. That was in the RTG 4510 mice. And also they hypothesized that this, in the picture to the left, a redox active ferritin, as you can see there, when it interacts with tau proteins, it can induce tangle of tau proteins, and and then cause neurofibrillary tangles. This redox active ferritin is believed it stores iron. As you can see there, there's iron in the center, and if this iron is redox active, this ferritin complex with iron can affect the tau protein. Then also they examined copper. So they saw that copper increased in the hippocampus and RT4510 mice, and the laser ablation inductively coupled plasma mass spectroscopy revealed that 79% of copper increase in the hippocampus and 61% of copper increase in the cortex. As you can see also in the right, the same picture that it was obtained with iron, they obtained it with copper. You can see the differences between wild type and TG. You can see here the copper bounded to the metalloproteins in both of the mice. Here they identified PIK3 as superoxide dismutase bounded to copper, and PIK4 they believe they could be other type of metalloproteins called metallothionein that are type of proteins that store copper. So the analysis of this copper bound to metallothionein, they found a decrease in copper bound to this protein metallothionein compared to in RTG4510 compared to the wild type. So in this case, copper bound to the protein was decreased with time. In the analysis of zinc, zinc alone, they saw a zinc increase in RTG4510 mice. The analysis revealed a 43% increase in zinc in the hippocampus and the cortex. In the analysis of zinc bounded to the metalloproteins, they saw 3 peaks. The third peak was the most important. That was the superoxide dismutase bounded with zinc. Also, I forgot to mention that superoxide dismutase is a protein that can protect against the oxidative damage. The zinc protein complex were altered in RTG4510 mice. This revealed a similar decrease in zinc bound to the proteins. So in this case, they hallaron a metalloprotein like ferritin in the case of iron, and like metallothionein in the case of copper, but they believed and other metalloproteins bounded to zinc are shown there, and they decreased as well as in copper. So in conclusions, these results support the proposed notion that a disruption in tau function may be associated with elevation in brain iron levels in neurodegeneration. This can result in oxidative stress and cell death, as the professor mentioned before. Ferritin redox active proteins, as I said, appear to induce a tau aggregation causing neurofibrillary tangles, but this is a hypothesis, so further investigation is needed. This study revealed changes in copper and zinc in the RTG 4510 mice, which are two important metals that have been implicated in tauopathies. There may be therapeutic potential in the targeting metals in disease that are characterized by abnormalities in the tau protein. And also, this is one of the first studies that to report accumulation of iron in the absence of amyloid beta. As the professor said, amyloid beta is another pathway that can induce tauopathies. Can induce neurodegenerative diseases. So this suggests that damaged tau proteins may contribute to increased brain iron levels in tauopathies.
So Javier, we're going to stop right there so we have time for questions, okay? All right, guys. Questions from the classmates. All right, so Omar has a question. I'm not clear on how
Yes.
ferritin could potentially induce NFTs if its general function is to store iron. Shouldn't it help reduce the elevated iron levels in the brain? Great question, Omar.
Yes, this is why I mentioned they had a hypothesis and further investigation may be needed. But if you can go, Professor, to there. So the hypothesis is that redox active iron stored in ferritin can make a conformational change in ferritin. So this iron binded to ferritin may become a redox active ferritin and thus inducing the tangle of tau protein if it reacts with tau. But this is a hypothesis. This is not well investigated.
Other questions? So I would think that it's not necessarily stored. It's probably a mutated ferritin that's probably leaking the iron so that the iron is exposed and accessible to possibly induce this. It most likely is not going to be stored within that's going to have the capability.
Oh.
Other questions regarding Javier's presentation? Adriana says, can we assume someone with anemia has lower risk of developing Alzheimer's? Javier.
I would say that it depends because this study specifically is conducted in a type of mice that has a propensity of developing tauopathy. So maybe in the people with the same type of gene as these mice that has a propensity of developing tauopathies, maybe an increased iron can induce the Alzheimer, but not necessarily.
Yeah, I would agree with you with that one because it's not necessarily the iron. In this case, is the loss of the control of the metal, and that's from the molecular mechanisms that are in place to regulate the efflux and intake of the metal. So in this case, that has probably more to do with it. All right, so let's go back. I know we're over time, but very quickly, Daniela had a question. No me queda claro si la diferencia o exceso de los metales es causado por el virus, this is COVID-19, o si es algo preexistente que causa que se empeore la condición de la persona. So if
Yeah.
Xiaomi or Alondra?
Yes, so we both have talked about this and have different opinions. So I'll share my opinion, and then Alondra can share her opinion. So besides age, sex, and pre-existing conditions, the article didn't present any other background information on the 306 patients, right? Therefore, I would assume that the majority of these patients had normal levels of metals and metalloids prior to their COVID-19 infection. There is no mention of the patients being exposed to high levels of chromium like the residents of Hinckley, California that we discussed at the beginning of this course. Therefore, my educated guess leads me to believe that these abnormalities in levels of elements occur due to COVID-19, although it is not sufficiently clear. Either way, one thing that both Alondra and I believe is that further studies are needed to answer this questions, since this is the first study that covers the topic of COVID-19 and levels of metals and metalloids. And Alondra can share her opinion now.
Okay. Alondra?
Sí, pues yo estaba discutiendo con Sayomi acerca de, o sea, yo pienso que el exceso o la deficiencia de estos metales es algo persistente, porque al analizar el artículo y otras lecturas como la de la nutrición y eso, ellos están hablando de que el nivel de metal es el que afecta cuán severo va a ser la enfermedad, o sea, el COVID. Y como que a mi entender, ellos te recomiendan que tengas una mejor nutrición para eso mismo. Incluso ellos mencionan que entiendo que es el hierro, que aumenta, la deficiencia aumenta el riesgo de que tú contraigas la infección. Así que yo pienso, aunque ellos en ningún momento especifican como que si esas personas estuvieron expuestas de alguna manera a los metales, pues yo pienso que es más algo persistente. Sin embargo, igual que dijo Sayomi, pues este es el primer estudio así acerca de esto, así que pienso que se deberían hacer más estudios y si acaso con una población más grande para poder entonces tener una respuesta firme.
All right, thank you so much. So actually, we're going to stop there, but I'm going to give everyone an additional question based on what your classmates have submitted to me. All answers of your questions go up online. Well, to some of the questions, I'm not going to give you all the questions. So you can always find them on the website, responses to some of your questions. All right, thank you guys. I thought you did a very great set of presentations. All right, and take care, everyone. Have a good break, mini break. Bye-bye.
Thank you all.